Um, Good day to everyone. I am Mariemme D. Martos, and I'll be reporting my thesis entitled Bioinformatics Analysis of Gene Targets for Birt-Hogg-Dubé Syndrome and Hereditary Leiomyomatosis and Renal Cell Cancer Syndrome associated with renal cell carcinoma using network analysis. So for the background of the study, RCC is a heterogeneous tumor that derives from epithelial cells of renal tubular which represents comprehensive 80% of all kidney tumors. Every year, there are approximately 338,000 new renal cell carcinoma releases in the world and about 30% of new renal cell carcinoma patients have metastasis at the time of diagnosis. Genes that are typically involved in RCC such as VHL, MET, FLCN, SDH, FH, TSC1, and TSC2 have an important role regarding with regulation of cellular metabolic processes. Most genomic alterations in RCCs are well defined until the WHO in 2016 discovered other classifications of tumors included in subtypes such as VHL, BHD, HLRCC, and HPRCC. BHDs and HLRCCs are major autosomal dominantly inherited syndromes. They are significantly syndromes because they are involved with the two common subtypes of RCC, which are the chromophobe RCC and papillary RCC respectively. Patients with BHDs deal with the RCC subtype chromophobe cell RCC. It is a rare autosomal dominant inherited disorder characterized by skin fibrofolliculomas, pulmonary cysts, spontaneous pneumothraces, and multiple RCCs. The germline mutation in the folliculin or the FLCN, a tumor suppressor gene, is responsible for this disorder. HLRCC are often associated with papillary renal cell carcinoma and characterized by predisposition to cutaneous and uterine leiomyomas and particularly form of type 2 papillary kidney cancer with an aggressive behaviors despite small primary tumor size. It is mainly caused by an autosomal dominant germline mutation in the fumarate hydratase or the FH gene. So this research will be examined through bioinformatics approach. In line of bioinformatic methods, there are three essential steps for analyzing databases. First is identification of significant genes or proteins, then network construction, and lastly, network analysis and visualization. To date, researchers have to learn to use a combination of several tools to accomplish this task, and interactive visualization of large networks has been primarily restricted to locally installed programs. Network Analysis was developed to address these challenges. It integrates all the three steps and presents the result via powerful online network visualization. So for the objectives of the study, identifying the main genes concerned in differentiating the pathogenesis on different classifications of RCC, specifically on diseases such as BHDs and HLRCCs, integrating the DEGs on web tool-based mainly named network analysis that will enable the user to construct protein-protein interaction network that will aid classification of the pathophysiological pathway of the subtypes of RCC. So for the data collection, the two different databases were gathered from the ARRE Express, which are EGEOD 11024 that includes 6 normal kidney samples and 6 type 2 papillary RCC samples or the HLRCC samples, and the EGEOD 21816 dataset that includes 6 normal kidney tissues and 6 Birt-Hogg-Dubé syndromes associated with renal tumor patients. These microarray datasets are inputted in text file and is uploaded in Network Analyst. For identification of DEGs, network analysis may also be used to distinguish DEGs between renal tumor tissue and normal kidney tissue samples. If one probe set does not contain homologous gene or if one gene has numerous probe sets, the data are removed. Fold change of the genes represent in BHD's tumor compared to normal kidney tissue and HLRCCs compared to normal kidney tissue were analyzed using Lima package. The comparison of interest is set with specific comparison to determine the genes that are significantly expressed in both datasets. The FD or adjusted p-values were kept to less than 0.05. Based on the fold change, genes were categorized into two classes, upregulated genes and downregulated genes. Genes that were normally upregulated genes or downregulated genes in both datasets were used for further analysis. Then, pathway enrichment analysis were performed to all common DEGs in both datasets. Network analysis allows users to perform functional enrichment analysis for highlighted nodes using different databases such as the GO, the KEGG, Panther, and Reactome pathway. So for network mapping, this step deals with constructing PPI networks. The summary level data from the two datasets are significantly altered in expression based on overall evidence. The significant genes of the datasets are presented in PPI networks and visualization analysis. In PPI network, the number of nodes, edges, and seed proteins are summarized for each subnetwork. As DEGs from both datasets were used to produce pathway enrichment analysis, therefore it conveys a much larger visualization in terms of connection of genes toward the disease. From reviewing the result in Table 4, it was interpreted that while the research of Much et al. May say that the chromophobe form of RCC have a lowered risk for metastasis, but its subtype, specifically BHD, showed that cytoskeleton protein binding has the highest gene count with 497 over 635 and has significantly lowered p-value of 1.39 e raised to negative 4, which means that a lot of genome that is involved in cytoskeleton protein binding pathway is included with the progression of BHD. This in turn may potentially point out that unlike the previously constructed views of BHD, it may possibly have higher risk for metastasis. Cytoskeletal protein binding contain different subfamilies of protein mainly which are microtubules, actin, and intermediate filaments. The mechanism of these proteins is altered in cancer cells as they promote tumor growth by increasing the cell's migratory and invasive function alongside its ability to proliferate and the resistance to cellular environmental stress such as mitochondrial and oxidative stress. Mutations from these genes may result in metastasis and because of the high genome count and the significance of its p-value, this may very well allude to the possible metastatic characteristic of BHD. In alignment with the statement earlier, three hub nodes were selected in terms of increasing betweenness, as this described the gene's centrality towards other genes that is involved in the disease's progression. These are KIT, ROB, UBC. The first gene is the c-KIT. c-KIT proto-oncogene is located on chromosome 4q and is considered to be part of class 3 of tyrosine kinase receptor. It is known to regulate several physiological functions such as hematopoiesis, erythropoiesis, lymphopoiesis, megakaryopoiesis, gametopoiesis, and melanogenesis. All of these are essential to the biological process of human beings. Numerous research have suspected that this particular gene could be a potential biomarker to the chromophobe type of RCC, as evidences show that if it is found 77% to 100% in cases of this type of variety and therefore is also potentially targeted for therapeutic modalities. Using network analysis, this figure shows that c-KIT is connected to a gene called RAC1. RAC1 is considered as one of the key regulators for cell motility and structure, as the members of RAC family is considered to hold regulatory function over cytoskeletal structures, mainly actin, as it primarily controls the mechanism behind the moderation of other signaling pathways that is involved in cell cycle regulation, cellular growth, formation of cell-cell adhesion, and contact inhibition, and these mediated activities are considered to be highly involved in progression of malignancy as it is included in angiogenesis, invasion, and metastasis, which are dependent from the mutation of the genes assigned in it. For ubiquitin C or the UBC gene, it is described as a stress-inducible upregulated upon different cell treatments as well as in other diseases. As it is one of the main hub nodes shown in the table that was detected through the use of network analysis, upon further inspection, WNK4, a subfamily of WNK protein kinases, is the one associated gene connected to UBC. Interestingly, recent studies about the link between WNK4 and ROGTPases have emerged. It shows that WNK4 can be isolated in a complex with ROGDI, As explained earlier, raw GTPases control the dynamics of the actin cytoskeleton and are important for cell migration and invasiveness. And while it is observed that the expression of WNK4 was found to be correlated with invasiveness as with the metastatic tumors such as infiltrative gliomas or in other neural tumor cells, the exact relationship between the genes is still largely undetermined. In addition, WNK4 is required for the activation of extracellular signal regulated kinases and mitogen activated protein ERK kinases pathways. Alongside other reports, this suggests that WNK4 is involved in many factors that attributes to carcinogenesis and has an important role in tumor cell growth and remodeling of extracellular matrix for tumor invasion. Results from enriched pathway from KEGG database from the Table 5 indicate that PI3-AKT pathway is one of the significant pathways having a gene count of 269 over 354 and has a p-value of 1.15 e raised to negative 4. A recent study shows that in PRCC associated with HLRCC dataset, PI3K-AKT pathway panel has 28% overall genetic alteration rate. The type 2 PRCC is associated with mutation of FH gene. Mutation of FH function leads to accumulation of fumarate resulting to upregulation of HIF alpha that mimics the loss of VHL in CCRCC. The VHL and PI3AKT pathways correlates extensively in a large signaling network. First, the upregulation of HIF due to VHL loss induces expression of a variety of growth factors including EGF, PDGF, and vascular endothelial growth factor or the VEGF, which activate the PI3K-AKT pathway through RTKs. This may aid in producing a further study regarding therapeutic target for HLRCC. In HLRCC, the hub nodes that has the highest betweenness is the CFOS gene, which is connected with the EGFR that influences the phosphorylation and activation of mitogen-activated protein kinase. The activation of MAP kinase is known to be the cause of phosphorylation of transcription factor P62 and ELK1. This can be attributed, therefore, to increased CFOS induction. This is especially significantly when talking about potential treatment for this disease. According to recent studies conducted by Gimeno et al., CFOS expression is regulated by intracellular signaling events and therefore can be treated as an important marker for EGFR, which have developed inhibitory drugs such as getfinib and erlotinib that are quinazoline derivatives aim to lessen the proliferation of assimilating defective cells by reversibly inhibiting tyrosine kinase activity. Furthermore, Mate et al. Reports to have observed positive effects of sunitinib and pasunib to metastatic type 2 papilloma RCC patients who have taken it for over 8 years. Sunitinib is a multi-targeted inhibitor of receptor tyrosine kinases containing a split kinase domain including VEGFR inhibitor. Sunitinib is under phase 2 in clinical trials, and molecular mechanisms underpinning sunitinib response and/or resistance remain unclear. Complete responses to RTK inhibitors are extremely uncommon, but in addition from the study, the researchers performed exome sequencing on the patient's primary and metastatic tumors, and they found out a series of potentially relevant genomic alterations including amplifications of the growth factor or the GRB2. Uh, suspected genomic of GRB2, which can be seen in the figure as directly related to one of the top genes, called KIT, was detected by network analysts and therefore strongly supports the accuracy of the program. This can be ascribed to the possibly effects of other quinazoline derivatives. In this study, RAC1, WN, and WNK4 genes were found to be connected to KIT, ROB B, and UBC. These genes are known to highly affect the cell metastasis of patients with BHD and plays a crucial role in cytoskeletal protein binding. In HLRCC, EGFR and GRB2 genes were associated with PI3-AKT signaling pathway that links with FOS and c-KIT gene that gave a baseline for new research targets for the treatment of HLRCC, such as common genes like UBC and KIT associated with BHD and HLRCC were identified but works differently in each disease. In conclusion, the present study has proven reliability of network analysis as a software tool that allows the user to perform interaction of profiling and gene expression data by generating accurate results that enable to correlate them individually to the given diseases. It gave significantly fresh insights for further examination on topics of diagnosis and widening berth of therapeutic modalities of BHD and HLRCC.